Hi, I have a little speech for you guys. So the topic is, what is gene editing? Imagine a future where genetic disease like scoliosis or Parkinson's disease can be corrected before birth, or where crops can live in very harsh environments. With gene editing, this is a very real promise of tomorrow. Today I'll be talking about what gene editing is and the ins and outs of it. Through countless biology and chemistry classes, self-interest in the revolutionary technology, and countless research on the topic, I looked up the latest advancements in biotechnology like CRISPR-Cas9 and applications in both medical and the agricultural fields. Gene editing is affecting our lives already and will play a very large role in our future through improving human health and our food production and our environment. I'll begin by explaining what gene editing is and how it works, and then I'll talk about the potential benefits in medicine, agriculture, and the environment, and finally I will discuss the big ethical issues surrounding the use of gene editing. All right, so first let's discuss what gene editing is and how it works. So what gene editing is gene editing involves altering DNA sequences to achieve specific outcomes. In this section, I'll discuss the tools and methods used in gene editing, including CRISPR-Cas9. Gene editing is a technique used to make precision changes in DNA of different organisms, so it can be used to repair and or alter genes to treat many things like diseases or specific traits. So CRISPR-Cas9 is a powerful and often used gene editing tool that allows scientists to cut and insert specific DNA sequences with extreme precision. The source is provided by Asmawa and Zawadi of 2021. CRISPR-Cas9 can be used to correct mutations that causes diseases like sickle cell anemia and beta thalassemia. This source came from CRISPR-TX website. CRISPR-Cas9 can also be used in agriculture with gene editing and help crops become more resilient to poor climate areas to help improve yields and stress resistance. And the source provided by Nduzu, Abigail, and all 2024. Other gene editing techniques and technologies like CRISPR are TALEN and zinc finger nucleus. All the same type of technology is used for targeted DNA alterations, which is all what this is happening. CRISPR is the more used platform because of how simple it is to use and how widely accessed it is. So, gene editing works by using molecular tools to cut DNA at a specific site, and then the DNA can be altered that way. CRISPR-Cas9 cuts the DNA at a specific site, and then you can insert, delete, or modify the genes that is being edited. Source by Asmawa and Zawadi, 2021. Scientists has used CRISPR to edit genes in embryos already to repair a genetic disease like cardiomyopathy, Cohen 2017. CRISPR is changing how we treat cancer by using precision cancer treatment, making it more targeted, effective, and tolerable. Source provided by Wang, Chen, and Wu, 2023. So, after cutting the DNA, the cell's naturally repair mechanism is used to insert or remove certain DNA segments through our natural body process. So, I've discussed with you that gene editing involves precise changes to DNA using CRISPR-Cas9. Now, I'll discuss the potential benefits of this technology. And now that we know how gene editing works, I'll discuss some potential benefits in medicine, agriculture, and the environment. Gene editing has potential for transforming different fields like medicine, agriculture, and the environment. While gene editing can change medicine by curing genetic diseases, it can also improve therapies and take on cancer. CRISPR has the potential to cure inherited genetic diseases as well by editing faulty genes. Treatment for people with beta thalassemia are also being done using this technology. Source from CRISPR-TX website. In 2023, Cassie VY became the first CRISPR-based medicine to cure sickle cell disease. Source by Henderson, 2024. Gene editing can be used to modify immune cells to better fight cancer, while research is developing gene-edited immune cells to target and destroy tumors more effectively. Gene editing can increase food security as well in different regions by making crops more resistant to disease, pests, and climate change. By editing the genes of the crops, we can create varieties that are drought and pest resistant. Sourced by Nzudu, Abergro, and al., 2024. Things like gene edited rice has used to prevent certain diseases and be able to withstand flood conditions, helping to solve for climate areas that are prone to flooding. Source from the USDA website. CRISPR is also being used to prevent certain diseases in tomatoes, apples, and mushrooms to help crop growth. Sourced by Synthgo, 2020. Gene editing can be used to protect endangered species and restore the ecosystem as well. In the future, gene editing could help bring back extinct species like the woolly mammoth. Scientists are using CRISPR right now to help de-extinct the Norway brown rat that has been extinct for almost 2.6 million years. Source Falter, 2023. Scientists are also in the works of seeing how they can de-extinct the Tasmanian tiger, as they have many tissue samples and the genome mapped out already. Source JANS, 2023. Gene-edited organisms could be designed to break down pollutants to help clean up the ecosystem. Researchers state in the near future they could introduce, swap, or remove genes to break down and detoxify environmental pollutants. Source ERS Genomics website. With advancements of CRISPR, we can engineer microbes that can produce bioplastics that can degrade easier and replace petroleum-based plastics. Source Meta Bioengineering website. Gene editing can offer growth in many different areas, from curing diseases to improving agriculture and restoring the ecosystem. Now I'll discuss the ethical concerns surrounding this technology. While gene editing offers immense potential, it also raises many ethical concerns that must be carefully considered. The rapid advancement of gene editing raises important ethical issues like concerns over the safety, equity, and potential misuse of it. Gene editing human genes, particularly in embryos, is questionable due to the leading concerns about designer babies and inequality. It is commonly known that in 2018, a Chinese scientist controversially edited the genes of two twins, causing for debate about the ethical issue. Dr. Victor Duzhou emphasized the importance of caution in germline gene editing, highlighting that its potential impact on the human race. Source Duzhou, 2019. There are concerns about getting informed consent, especially with editing human embryos or doing clinical trials as well. Gene editing in wildlife and plants could have unintended ecological consequences as well. Exposure to CRISPR/Cas9 pesticide could unintentionally alter the genes of an assortment of non-targeted organisms. The metabolic enrichment analysis confirms the potential of unedited effects to cause adverse effects in non-targeted organisms due to outdoor gene editing. Source Hopers, Heinemann, and al., 2024. As gene editing technologies become more accessible, there's a need for strong regulations to prevent misuse. While gene editing presents incredible opportunities, it also brings ethical dilemmas and risks that should always be examined. Now, as I begin to summarize, it is important to note gene editing is very exciting and scary all in one. Gene editing is a powerful tool that has potential to change medicine, agriculture, and the environment. It also raises many ethical concerns about how it can affect human genetics, biodiversity, and safety of it. As gene editing is at the beginning of changing our lives, we have to remember how far are we okay with these advancements and what kind of future do we want to create. How we use this amazing technology will shape the world for generations to come. Thank you for listening.